So, so I think with this, I would like to welcome you all to this webinar. My name is Rasmus Bro from University of Copenhagen in Denmark. But more importantly, we have Christian Zachariasen, who has been working with what I used to call process chemometrics for many, many years and in many different places. Nowadays, maybe it's called process analytical technology, and there's probably even a more fancy word for it that Christian will talk about. But he will talk about how to introduce analytical methods in production and associated problems. So, Christian, please. Oh, sorry. Let me just mention, if you have any questions or comments, please put them in the chat, and then I will convey them. Thanks.
Rasmus has promised to be a moderator, and if you have any questions, please write them in the chat, and Rasmus will moderate them, either interrupt me or keep the points to the end. And I think at the end of my presentation, we will open for everybody to unmute and have a discussion, and please chip in with that discussion. The topic of my presentation will be pharmaceutical method development with the focus on early stage development. So going from idea to sort of having a mature method ready for validation, pharmaceutical validation, and the detailed workflows as we see it here in our department at Novo Nordisk. It is not going to be how the pharmaceutical regulating bodies want this done, or all the crevices in that, but actually how we do it at Novo Nordisk. And I will go through the principles as we see it, but for reasons of confidentiality and so on, I cannot share any practical examples. And that would also go a bit too far, because I've at least themed this presentation to be about the overall principles. My background is I have a Master of Science in Chemical Engineering from the Technical University of Denmark. And then I've been working sort of like with PAT and chemometrics before PAT was invented. But from 97, when the process analytical chemistry term was coined, and it was picked up by FDA in 2004, I think. And then I've been working in various food ingredients, consultancy, and biotech companies, also a short spell in the dairy business, before landing in Novo Nordisk in 2020. So I've been sort of covering everything that a good scientist needs to do, and not just something doing data science, but actually also sampling, quality by design, manufacturing intelligence, DOE, spectroscopy, batch modeling, process modeling, all the sort of things that a real scientist should do, rather than people just labeling themselves data scientists because they can use, let's say, data mining models. So the question that I will try to answer is how to do analytical method development in a pharmaceutical settings, at least how we do analytical method development in Novo Nordisk, with focus on the journey from innovative ideas into validated, controlled, and documented methods implemented in a laboratory setting or process environment. And if you look towards the governing bodies, one example of how to do analytical procedure development, you can ask the International Council for Harmonization of Technical Requirements for Pharmaceuticals for Human Use, that is the full name. They have this Q14, which is depicting the analytical procedure life cycle. And if you look at that figure, that is one of the most confusing figures I have ever seen, because where do you start and where do you end? There's a lot of arrows there going around, and it, of course, is exemplifying that analytical procedures are often an iterative approach. You have to bring in a lot of elements, and you have to keep improving, continuous improvement, and also revisit things. So at least it will try to incorporate product and process understanding. I'll see if I can find a laser pointer here. So it incorporates product and process understanding. That is also what you can perhaps put in here in the prior knowledge box. It would like to have some ideas about control strategy coming from the process. And then it mentions an analytical target profile, which is actually what you want to measure. And then it goes into technology selection and risk assessment. And here you have some things, parameter set points and ranges that should actually be covered in the analytical target profile and so on. And yeah, so this is, let's say, a top-down view of an entire pharmaceutical business trying to evolve. But it's not. It's hard to get a very structured approach to let's invent something new, because where do you start in this circle? So we have tried to make a more boring or linear interpretation of the workflow, which indeed is iterative, but you are able to go back and revisit things. So what we would like to do at Novo Nordisk is to when we have an idea, to establish a proof of principle. And I will go through all these, at least I'll go through the green steps in this presentation in more detail. So we would like to establish a proof of principle. That is when we have an idea and have some task that we want to do, how can we solve this task in principle? And that means if we can get instrument X to measure Y, then we have established a proof of principle. So that is a very simple way from idea to possible solution, at least outside of a regulated pharmaceutical environment. And if the proof of principle shows that we have somewhere that we can go ahead with, we want to take it to a proof of concept. And proof of concept is more internally to the problem at hand and a specific solution working in a pharmaceutical environment. So now we're looking into not only using a instrument, but also which instrument should we use, what are the requirements for that, and how do we establish something that we can push further on into our own internal process. And then when we have established a proof of concept, we go into basic method development. So we get a lot sharper on what we want to do and how we will achieve it. And that will form, if that is still successful and we still have a go, everything that we do in method development should point us towards how to validate the method in a pharmaceutical settings. And there are a lot of strict requirements for a validated method in a pharmaceutical setting that ensures safety of the patient and so on. And then when you have a validated method working at our labs or at one process line, you need to transfer that method to other sites. And there are Then ways of doing that, ensuring that the validated method that you have established is also transferred correctly with instrumentation and method and training and capabilities. And then there's method support, and we go into method LCM, which is an abbreviation of life cycle management. There will be a lot of abbreviations in this presentation, and in Novo Nordisk, we often forget how many concepts we abbreviate into 2-letter abbreviation, 3-letter abbreviation, or perhaps 4-letter abbreviations. So, and then we just throw around with abbreviations and we forgot what they mean. So I will try to explain all abbreviations that I use, at least once, and then we can get back to that. But LCM is life cycle management. So that is how you maintain the method and ensure that the method is controlled. But I will focus on the first 4 arrows of this until we have a validated method. So the ideation and or proof of principle stage, which should read POP rather than POC. I can see the headline should be POP. The proof of principle established in principle that an analyte can be measured with a suitable technique, solving a tangible problem or challenge in the process or accompany pain. And for in this stage, the measurements can be made internally at Novo Nordisk, or it can be made externally at a vendor or at universities. When you go to conferences, you often meet vendors that say, we can measure this and that very smartly, so just send us 20 samples that have problems to you, and we will measure it and give you a solution to all of your problems. And that can be a good way to try out new ideas and concepts. We also have lots of collaboration with universities, and we have lots of ideas being developed in collaboration with universities. So everybody is allowed to get ideas, and then if you have a good idea, by no means keep it from yourself. Pitch it to the team. We would like the idea to look into some kind of company pain. So it can also be that you meet colleagues that says, we really have problems controlling this or that, or wouldn't it be nice if we could measure this or that, or something like this. So either we have a pain that can give an idea, or we have an idea for a pain. And that is very much about internal networking in your company. So when we have an idea and a pain, and that is matched up, we can start going tech scouting. So what can be a possible solution to the pain from an analytical standpoint? And then we need to scope what area is the idea in, what suitable instruments are we looking to solve a problem in a QC lab, or are we looking for a problem to be solved on the process line, or are we wanting basically to do both? So establish a lab method and then push that lab method into the process. That can also be a scope that you look into. So what do we need to conduct a successful first experiment? So you need to narrow in what the pain, does it translate to some concentration of an analyte? What is the matrix you want to measure it in? Are there any possible interference, or are there any, if you want to measure inline, are there any problems establishing a measurement point? And then we can set out and do a simple lab scale proof of principle experiment, either at vendor or at ourselves or at the university. Then, and that can be 20 samples with varying concentration in a particular matrix to see if that is suitable. So do we get usable results out of that? And those results or data, does that appear to resolve the pain that we had? If not, maybe it's time to get a new idea or a better idea, and maybe you learn something that you can bring forward. But so that's an iterative cycle. But once you think you have a solution to a pain and an idea to how you want to solve that, you present this to the team. So I have measured A in this matrix using this instrument, and the first experiment looked really good. Then you can move into the proof of concept phase, which has an included equipment sourcing, because then we would like to internalize what we have maybe done in outside of Novo Nordisk, or what we have done with just the instruments that was available by chance or by friends of friends here at Novo Nordisk. So we want to get that step further, and we want to be more sharp at the scope for the method and get more sharp on the requirements to the instrumentations. So the proof of concept should be made at Novo Nordisk using Novo Nordisk materials and in a relevant Novo Nordisk matrix. So matrix, let's say, solution that the analyte is within that pain that you want to solve. So if we did the POP within Novo Nordisk, then we have an idea about the equipment. It could be that we need better equipment. So if the equipment is available, we can proceed. But if the equipment is not available, for instance, a vendor we met at a conference, we have this very smart new instrument that is much better than everything else on the market. We can consider if we need to buy that instrument or an alternative capable instrument is available. So scope, equipment, and price, make a user requirement specification to the instruments to the best of our knowledge at this stage. And then when you have to spend money, you have to tell management, I want to spend this money, but we have this really big pain that we can solve. We have a great idea for that. So make a basic business case and investment application, and then buy the new equipment. And often equipment comes with a PC, a computer to control the equipment if it's not built into the equipment. And then if we need a PC, then we are starting getting some sort of pushback from our pharmaceutical organization, because Novo Nordisk would very much like to control the PC that goes on the Novo Nordisk network because of IT breaches and problems and regulations. Not because the individual PC may be at risk, but that PC could bring other PCs, other computers, other network at risk. So either we basically have to bring the associated software needed for the instrumentation on a Novo Nordisk approved image if we want the PC to go on the network, or otherwise we can do what is called a standalone. And then if the supplier or vendor supplies a PC that we cannot control the image of, then we can have it, but it cannot be connected to the internet, and we have to use it has to work completely offline, and that's a pain when you want to update software and so on. but then that can be brought on with virus scanned USB sticks and all that stuff. So either we need to clarify if we are working offline with uncontrolled PCs, or we want to bring in a Novo Nordisk PC image and have the benefits of a network for remote control, for data transfer, and so on. There is a lot of trouble. It's just need to go to with a USB stick and scan every time. So it's making use of the instrument a bit more cumbersome. And then you basically receive and set up the new equipment, including a computer, and then you arrange what is called IQ, OQ, which is installation qualification and operational qualification. And that's an agreement with the vendor of the instrument that they deliver what we think we are buying. So if the vendor promises to measure A, B, and C, and the specification of the instrument is A, B, and C, then we actually have some documentation that the instrument have the performance as promised. Most often, we would like to engage with vendors that understands pharmaceutical requirements and can deliver IQ, OQ vendor documentation, and then we will review it and see if it is sufficient. But if not, then we are at least pushing a pain in front of us in the later stages. So we have 3 levels of equipment. So when we have equipment for tests, we have basic requirements that they should be safe to operate. And here in Europe, we would like the instruments to be CE marked, so you don't get electrocuted by touching the instrument. It should be properly grounded. It should be safe to use. If the instrument, for instance, has lasers, we would like to review whether you can get blinded if you open the wrong port on the instrument. So basic requirements to safety and operation of that. IT security has to be handled. As I said, if we have a vendor PC that is uncontrolled, it will not be allowed on the Novo Nordisk network. And we can use that for test of equipment and proof of principle. And we don't have a lot of requirements for documented training and so on. When we move into proof of concept, as I just covered, and we use it to evaluate if we can build models relating the data from the instrument to the analyte that we're interested in. And for method development, we would like the instruments to conform to what we call GSP, and that is good scientific practice. And the loose interpretation of that is that the instrument can deliver measurements that we trust. So that means we need to have some sort of calibration and some sort of performance confirmation. And that is usually that can be established by the vendor IQ and OQ. So what is the baseline performance of the instrument and how is that controlled? So if we're not doing that in the POP phase, we will need to address it in the POC phase, because we would actually like to base our decisions on measurements that we can trust. So we need some sort of performance confirmation and calibration on the instrument. And perhaps if we trust the instrument and the vendor, maybe we can say we want to check that on a yearly basis. But at a later stage, we may need to test the equipment much better and much more often to ensure that the instrument is in a validated stage, but that's a later pain. We also need for GSP equipment to establish O&M, which is another abbreviation, that is operation and maintenance, to make sure that when we have GSP equipment, that it stays in GSP state, that we can continue to trust the measurements. And that is hard to do if you don't maintain the instrument. So we can have a nice instrument conforming to GSP requirements on day number 1, but if we don't maintain the instrument as it should, if we put in wrong liquids, if we don't follow the guidelines set forth by the vendor, the instrument will go out of GSP control. Also, we need a guide on how to operate the instrument, because if we don't operate the instrument in the right way, if we expose it to thermal shocks or liquids that is not pressure, that is not working for, then the instrument will also go out of GSP control. And for that purpose, we introduce logbooks at this stage, where you should at least notify everything you do that could change the performance of the instrument. So this is not the full logbook where you need to document everything you do on the instrument, but at least everything that has an impact on the state of the instrument. So for this, we start having requirements for training for people who use the instrument, and how to maintain the equipment. And then when we want to have validated methods, we need qualified equipment. So we use this terminology in Novo Nordisk, that we qualify the equipment and we validate our methods. So that is the distinction between qualification and validation that we make. So for GMP requirements, we have all the requirements for GSP and below, and then we need to go through a full qualification package. And that means both the documented installation qualification, operational qualification that we mentioned in the previous stages, but now we're also adding in checks to see if the instrument is in control. So that can be what we call a PQ, a process qualification, that the instrument is in control for the intended use. And we actually would like to test that the instrument delivers the expected performance every time we use it. So we can have, before we run any samples, for instance, we want to do a system suitability test, an SST, which means that we, for instance, run a known standard and get the expected result from that standard. And that standard should reflect the use for the instrument. So in order to validate a method, we need GMP qualified equipment. So we need that for method validation for all GMP samples, so all method that we release for patients, for stability studies, and also for the studies when we want to push our method out on to other sites, they also need fully qualified equipment. And then we have further requirements for training in equipment and methods and so on the GMP level that we need to operate it on. And furthermore, if we do changes to instruments setup or to method setup, we need to have change control documentation. We need to push that through our quality assurance. And if we see anything unexpected going on at the instrument, we need to write deviation reports. So a lot of papers to ensure that the instrument and method is in control, is continuously in control, and there's no risk to patients, because we don't really want patients getting sick of taking our medicines. We want to cure patients and not make them sick. So these are the requirements to the equipment. So going back to the method development on the GSP level, so that is for the proof of concept. We would like to write an analytical target profile that was also mentioned in the ICH Q14, which states the purpose and requirements of the method. And the ATP is sort of the document covering, let's say, the negotiation between us that are developing the analytical method and the customers. So people that have the pain, what pain do they need to have solved, and what are the requirements for the analytical methods? So that's a negotiated document where we agree. We have very specific requirements to what matrix is the analyte in, and what are the levels that we need to have the analyte assessed in. What are the how do we sample? How do we take out reference sample for secondary method? Everything that is relevant for method development, let's say the negotiations should be part of or included in the ATP. And then we need to write method protocol for which experiments we would outline, how to measure which samples, which samples do we present to the instrument, and how do we make sure that these samples, for instance, mimic process samples or product samples. If we are measuring QC products, let's say finished products in the QC, then we can take samples from our QC, but more often we need to make the samples ourselves because we need to span out the variation. So if we have, let's say, predefined concentrations in the QC, we definitely need a method that can work outside of the existing products to span out the variation. So we should make an analytical target profile with the stakeholder and write as an experimental method protocol, and then get somebody to review and approve that. So the stakeholders should review how we are going to measure our samples, and then we start introducing, following the protocol, measuring the samples on the instrument, and likely we need to optimize the method and instrumental parameters. So we conduct experiments, collect data. We do some geometric model building on the data that we collect. We need to challenge robustness of what we are doing, so we are not just lucky or just are making an overfitted model that is not robust. So we evaluate this, and if the criteria that we have set up for method robustness is not met, then probably we need to optimize the method, both instrumental and let's say protocol-wise. But once we are satisfied with the method as it is, then we consolidate our findings in a method development report, and we present and have that reviewed with our peers, and then we evaluate if the POC, the proof of concept, is fit for purpose, and then we can push it into the next stage. Are there any questions so far, Rasmus?
All good here. Thanks.
Okay. If you have some questions, please speak up. Otherwise, we will push into GMP method validation. I will do so. So we need, as I said, qualified equipment for a GMP level investigation. So after a POC, we want to push into final method development. So we need the equipment to be GMP qualified, and we need all associated IT to be GMP qualified. So in order to do that, we need to make sure that the qualification of the instruments ensures that the equipment is calibrated, in control, in compliance, and performs as intended, and operated according to all internal and external standards that we foresee we are going to use. So on the IT side, we need to write an IT risk assessment, so ITRA. So we need to evaluate how the software controlling the instrument is working. And we have to write an ITOM, so IT operations management. We need to write very specific guides on what the user and method responsible should do in terms of operating the software. Because now if you change things if you have a method that works on an instrument and you change the settings of the method, then you are changing how the instrument works, how the instrument acquires data, and that will jeopardize, in principle, your results and the operation, the data that you get. So based on that, we should write user guides and control standard operating procedure on how to take measurements on the instrument. And bring that about, and then you write a qualification protocol, which again get reviewed, and then QA will review this. Most often at this stage, we will start giving user roles, so there can be a user of the instrument which is entitled to take measurements on predefined methods. We can have a method responsible that is responsible for setting up the method, but that method should be locked for the user, so the user cannot access the method and do changes to the method. And also the method responsible should note any changes to the method and see if that impacts how the instrument acquires data. So, and if there's changes needed on the methods, we have the whole change request case. We want to change this in the method, and we need a risk assessment on if that will impact the state of the instrument. So we run an IT qualification that the IT is safe and sound, and then we run equipment qualification that based on the IT software that is qualified, then the instrument is also qualified and is meeting those requirements. And then we need to write a qualification report that gets approved. We have very strict requirements to, for instance, data flow on GMP level instruments. For instance, the authorities, they would like to check that that there's no risk of us intentionally or unintentionally manipulating the data after acquisition. So for instance, if we produce data, they should be backed up to a read-only data drive that is controlled, so that nobody can edit the data after it has been acquired. And we actually need to back up our data within an hour at the latest. So if we are collecting inline data, let's say on a 6-hour process, we should actually terminate data acquisition after, let's say, 55 minutes, write the data and start up a new data acquisition. So for instance, if the power is if there's a power cut, that we don't lose data. Also, we need to prove to the governments that if we, for instance, measure a result that we don't like, then we don't have the opportunity to kill the computer. And this is, oh, that was such a bad measurement. I cannot live with that. I don't want to write a deviation report. So I just pretend I never took that measurement. So these are some of the requirements to measurements on a GMP level. So we have and also if you acquire some data and then 2 weeks later you open them and model them, and then maybe you have opened some spectral files in a CSV data, and then you intentionally have a cat walking on your keyboard, and adding some extra characters to your data file, and you get to save that, then you have suddenly manipulated data unintentionally, but that could also compromise patient safety. So there's a lot of requirements to GMP qualified equipment.
Christian?
Well, yeah.
There's a question related to something you said in the beginning of the slide about when you change the method. How does that relate to, is the question, how does that relate to issues like instrument drift?
That is very often, that's a very good question, because when I am developing a method, I am doing a lot of tweaking of the software. Maybe we should have a gain of 2 rather than 1. Maybe we should try 128 scans rather than 64 scans. And we are playing around a lot to optimize the method. But once we have a validated method, we have a locked. We also have a method on a low. I mean, when we have let's say a validated pharmaceutical method, that goes hand in hand with a validated method that we use to generate the validated pharmaceutical settings, and that method is considered to be locked. So if we are changing things in the method, we need to do a risk assessment, whether we need to revalidate the method, or we need to, if we can, let's say, write a rationale that those method changes that we do is okay. So getting back to instrument drift, if we during early method development notice that the instrument is drifting, we should make then we would clearly see here that our method is not robust, because the results we get after 4 hours does not look at all like the measurements we got when we just turned on the instrument. So we need to address that in the method. So a method could be turn on the instrument, wait for 1 hour until the instrument is stable before you can start taking measurements. And then you can start taking measurements. And then you should start taking a background, for instance, a background spectroscopic scan for what you are doing. And then we should evaluate how long time is that background valid. Is it valid for 1 hour, for 9 hours, which is in practice to the end of a normal working day? Is it does the background expire after 25 hours? So the next working day you have to take a new background, or is the background good for a year? That is likely not possible, or not very good for at least a validated method, because if your background 1 year after is deviating, maybe you have put your patients for a full year of production at risk. So when a method is robust, it needs to be robust on all levels, including instrument levels. And you need to incorporate how to mitigate instrument drift into your method by, for instance, taking background, and you need to make sure that the background expire after the requested time period. So that is a requirement, that if the software supports the expiration of a background, then we can just write in the software and say after 1 hour you cannot take more measurements, you can start more measurements. If the software is not very GMP ready, then we need to have protocols establishing that humans should ensure that this is not the case. And then you need to log when did you take the background, and then for every measurement, which background did I use? And then you need a reviewer to look that the human that took the error does not have that in a valid background. So if the software does not help us enforcing GMP rules, we will have to add human reviewers to exclude the chance of getting an error of taking a measurement with an expired background. And that's a lot of work and a lot of manual review and a lot of documentation and a lot of logging and a lot of, is this time, this spectra, what background was the timestamp of the background of this sample spectra that I took. So we would really like the software vendors to write pharma and GMP compliant software. That's a lot of work, especially if you are grabbing the newest idea on the shelf and you have a startup company and they have made 10 instruments and they have outsourced writing the instrument software to some guys in India who can make it for $10,000, and then it's working, but it just collects data from the instrument. That's not suitable for GMP qualification. Did that answer the question?
Yes, Neil said thanks.
Cool. So when we then push method into validation, we need to, everything that we do on the instrument side, we need to ensure is also working for the method. So the method validation ensures that the method is in control, in compliance, performs as intended to all these standards. And then we can have requirements to accuracy, precision, specificity, detection limits, quantitation limits, linearity, range, and robustness. That is all mentioned in ICH Q2, which describes method validation. And some or all of these requirements can come into play, but you can also have, if you have an ID method, so is this analyte present or not present? For instance, then you may not have a requirement for linearity, because we don't need to give we don't need to tell how much of the analyte is present. We need to have a detection limit. So if we are above the detection limit, we have a confirmation that the analyte is present. We have a positive ID. And if we have a negative ID of the analyte, we know that it is below this required content. For instance, 0.5%. If we have a result that is below 0.5%, if that is the limit of the method, that could be sufficient for an ID method. So all methods do not need requirements for all these things, but let's say quantitative methods will likely need most of them. And we need to ensure that this is valid not only when we let's say, validate the method and say, now it's ready for production. We also need to ensure that every time we take measurements going forward at all sites, all over the world, with all equipment and all operators, that these requirements are met. So we need to prove that we conform to the validated performance of the method at all stages. If not, if we notice a deviation, then we should record it, and we should immediately do an impact assessment. So this instrument or this method is out of control. When was it last time documented in control? What measurements have we taken since we last knew it was in control? And do we need to quarantine or to recall or remeasure these things? So worst case, the products could be on the market, and we need to do a recall, and that's everybody. That means we need to tell everybody in the outside world, hey, we made a mistake, we think, or there's a chance that we made a mistake. So we would like to recall the last 3 batches of Ozempic. And then we hope people have not bought the medicine and injected themselves, because we put patient at risk. So that's why we have a release control of all products before we release them to the pharmacies. But those release methods based, of course, on the validated methods. So we put patients at risk if possibly we put patients at risk if we have a deviation. So that's why we take our deviations very seriously. And the deviation is something is not behaving the way we would like to see. So in order to validate a method, we write a validation protocol. So that validation protocol is based on everything we know until now. So how can we make sure to address everything we know from now? How do we want to measure this analyte? We need to take into consideration instrument robustness, method robustness, variation in the matrix, and all these things. So we really would like to see, we want to measure these 40 or 80 or 200 samples with this variation, with these changes in temperature and pressure, and with these small deviation in instruments to make sure that the instrument is robust. So we use DOE a lot to span out the samples that we use for our validation protocol. And then that is scrutinized by scientific peers and QA. And then from the protocol, we prepare test plans, which is now, let's say, the validation protocol will say we want to measure these 20 or 40 samples. A test plan will say these 40 samples needs to be measured. We want to name the measurements in this way so we can have full traceability of what we say we measure and what we actually measured for later modeling. So again, these test plans are reviewed. Then we need everybody to be GMP trained in the operation and maintenance of instruments and the method and how to handle the samples. And then we have, so they know all the bits and pieces in the standard operating procedures. And we don't just ask them to read an SOP, we actually also want them take them to the instrument and train them physically at the instrument so we are sure they understand the SOP, the standard operating procedure for what we want to do. In the case that we are doing simultaneously, let's say PAT measurements with a reference method, the protocol will also outline how we should handle samples for reference samples. Let's say if we have a reference sample, it could be a UPLC method. We would call that the primary method, and the PAT method is the secondary method. So we want to validate that we going forward can replace the primary method with the PAT method. So in that case that we have the dual sampling, the reference analysis for the primary method and along with the PAT sampling, that needs to be covered in the test plans. And of course, we would like to if it's possible, to scan a sample using our PAT instrument and then make sure to send that exact sample to the reference measurement for the UPLC. But then, for instance, we need to review sample handling. So, for instance, getting an assessment of water content is notoriously difficult because the analyte is all over in the air. So how do you ensure that the sample you measured on your at-line method, that the water content does not change while taking it out of the instrument and into the lab for Carl Fischer water determination? That is nearly impossible. And when you have online or inline methods, extracting the samples that pass through your inline or online sampling cell, that is impossible. So then again, you need to write a good rationale why you that describes that you're not cheating that much when you are using sending these samples to the lab for reference measurement, and you actually scan something else in your inline with your inline probe. And there's different ways to mitigate that. For instance, instead of going inline in a full-scale process, maybe you can develop a small-scale process where you can, instead of doing samples from a large blender, you can make a small blender and then measure on that and take everything in the blender for reference analysis. So that rationale between the PAT samples and the reference samples is extremely important to when developing a method that you later want to claim to be valid. If you need preparation for your reference method, you should make sure that this is the place. So for instance, if you want to compare your new PAT method with a UPLC method, you need to make sure that the UPLC method is there and that it's also validated, because we cannot claim to have a validated PAT method if the modeling we used for the PAT method was a reference method that was out of control. That will give us big problems. So if there are requirements for the reference method, we need to make sure that these are met. But then in basically you run your PAT samples, you run your reference samples, and then you write your validation report and you validate that. So it's very important here. One very important aspect of this is that when you do method validation, you have actually completed your method development, because what you are validating is the method as you said it would run. So, for instance, you part of the validation for the PAT measurements, you already have developed a method, and you could say you are actually testing the performance of this model, multivariate model, because you just bring the model into your analytical system, and then you test if that model will give the expected results proven by the reference method. So if you could say that this is the final test set of a method, and if you have no possibility of doing variable selection to fit your PAT measurements to the reference measurement at this stage, you have no possibility of changing components. You cannot change instrument settings and so on. You are validating the multivariate model along in this way. And then you can say that model is released, but and then you're good to go. But you're only good to go on the instrument that you actually brought into testing. So you need to then, on the backside of this, you need to do method transfer. So typically, if you do your method validation on one side, you need to look into instrument variation. How is that handled, both on the PAT side and on the reference method side? So if we should test several PAT instruments to make sure that the instruments are robust, or you should build method or instrument to instrument variation, possible instrument to instrument variation into your method. So for instance, you do a background and you do a calibration, you do some sort of a background correction that will work on several instruments. And also your reference method needs to work on several of reference instruments, so several UPLC instruments. And that's basically what you test in method transfer. I'm not going to all the details of this, but basically you take a small subset. Usually you can do with a small subset of the samples you use for validation, and you want to rerun that on another site with new instruments, new operators, new reference instruments, and you want to make sure that the PAT instruments and the operators and the reference instruments are all in compliance. So there are lots of opportunities that a validation can fail, and then you have to go back and make your method more robust, or the SOP more precise, or whatever caused an unexpected result or the failed validation. You were simply not, you didn't mitigate all the risks in your method. So then you are back to the let's say the POC, and often we have a phase beyond POC where let's do the method from, let's take the method from the POC method until we have a more robust method ready for GMP level. So that is the Novo Nordisk take on what work packages that we need to do. There's obviously tons of details into what we put into the different boxes, but that is beyond the scope of this this overview. For our work, we have identified a lot of guidelines and technical standards to look further into this area. Some of them are specific for near-infrared spectroscopy, which you could say the authorities are more acquainted with those methods than newer instrumentation. But the principles are the same, and the considerations and for instance, instrument-to-instrument variation that we see on near-infrared spectroscopy can readily be applied to other types of spectroscopy with plus or minus some extra additions. If you want to do Raman spectroscopy, maybe you should look into the laser and the risk about that. But then the laser is replacing the light source of near-infrared spectroscopy. So there are two specific for near-infrared spectroscopy, but very detailed regarding that. So those are very useful, and they have lots of sound principles to transfer to other instrumentation. And then there are 6 guidelines and standards, which is a good start. It's by no means an exhaustive list, but those are good starting points on sampling, on multivariate analysis, and on method development and instrument control.